Hello, everybody, and welcome back to another episode of Eyeing Progress here on the Curing Retinal Blindness Foundation YouTube page. I'm your host, Mitchell Smedley, as always, speaking with a tremendous person in the field of vision research. And today is no exception. I have the one and only Dr. Dan Chung on the show. Thank you so much for joining us, Dr. Chung. How you doing today?
Great. It's great to see you. And I am just super pleased to be with you today. Again, apologies for the delay in getting us together, but very happy to be here today.
Not a problem at all. It's great to be seeing you and not having to sit in a room alone in the dark for 45 minutes.
Yeah,
We'll get to
we
that in just a few minutes. But
absolutely. We appreciate all the support that you've given us in the past.
it was fun and it got me out of school. So, for
There you
those that
go.
don't know, I was able to, I had the great pleasure of meeting Dr. Chung way back when I had to be what, like 10 years old, something like that.
Yeah, you were a
Um,
little guy then. Yeah,
yeah, still am, but nothing's changed. So, yeah, and
not
we
seven.
I guess I'll start even before that. Where did you kind of get your start in vision research, and then we'll kind of build throughout your career, and then we'll talk about how you and I met. So, where did you first get your start?
Yeah, for me, I trained as a pediatric ophthalmologist, and during my fellowship, there was quite a bit of ocular genetics and inherited retinal diseases. And it just turned to the idea of getting into some clinical research and some basic science research. And I was fortunate enough to get a position with Dr. Jean Bennett at the University of Pennsylvania, who was working on some really neat stuff in the field of gene therapy, which eventually led to the first gene therapy to be approved by the FDA and one of the first for the EMA. So that's really how it all started. I was at University of Pennsylvania for 12 years working on inherited retinal disease in the lab and then in the clinic or doing the clinical trial for the RP65 program. And really, even before then, I did a fellowship at the NIH in retinal gene therapy. So I've been in the space, for a little under 30 years, minus the years for training. So it's basically all I know. So, but I do love it. Absolutely.
That's a long journey. That's incredible. 30 years. Congratulations, and thank you for all that you've done.
Well, it just shows you how old I am. So
Well, for what it's worth, you don't sound a day over 30.
I'll take that any day. Thanks so much.
No problem. So let's kind of jump forward in time, I guess. So it was a little over 10 years ago. So I guess 20 years into your career, kind of a big jump, but whatever.
Yeah.
You were working on a project, and that's where I came in, I guess. I was helping out running some obstacle courses for you. So why don't you tell everyone kind of what that was for, and why you enlisted someone like me as your little
Well,
lab rat
absolutely.
there.
And it was a key thing that you and your brother were involved with is that for the RP65 program, there's not a lot of endpoints that have been used in phase 3 trials in inherited retinal disease clinical development. And just because there haven't been a lot of phase 3 trials, and we realized that visual acuity, which was very common at the time as being the endpoint that the FDA really liked to see in ocular clinical development, we felt that that wasn't the best way to show that the product itself had a beneficial effect on those who were treated with it in the clinical trial. And basically, this was the brainchild of Jean Bennett and Al McGuire, was to see if there was a way that we could show that after treatment, folks could navigate an obstacle course under dimmer light conditions, because one of the first things we see in RP65 disease is the loss of light sensitivity. And so we devised this mobility course, and this was for the primary endpoint for the regulatory trial, the phase 3 trial. And we had to run a validation study. And what we incorporated with folks that had inherited retinal diseases and those that didn't. And we would just compare the results after 1 year. And so obviously, we were very happy that you were willing to participate in doing that and kind of giving us an idea of what it was like for folks with inherited retinal diseases to try and navigate the course itself. And so it was very instrumental in getting the course validated, and eventually it was used in the pivotal trial. And it was really based on that data that we were able to get the approval from the FDA. And in the long run, the videos were very informative of before and after for our clinical trial participants. So great kudos to you and your brother and to the other folks that participated. We're very grateful for that. So
Well, I'm glad I could be of help just, kicking through a bunch of your wooden blocks and whatever else there was placed out there. I hope that was inspiring for everyone. So,
it was, and it's still on video. So if, we'll have to talk to, Chop or Pen or Spark, see if you want to see those videos again.
I don't know
So
if I want to relive that. I can't imagine I was very controlled back then at 10 years old. If I couldn't figure out the obstacle course, I probably
you did
stormed
great.
out of the
You
room.
did great. No,
It was
nothing like that.
ridiculous.
Not at all. Not at all. You did fantastic. So.
Oh, well, thank you. So that was for you said that was RP research?
Yeah, that was for a disease specifically called well, basically it was inherited retinal diseases associated with a biallelic variance in the RPE65 gene. And this led to an early form of childhood inherited retinal disease. A lot of times it's called Leber congenital amaurosis or early childhood RP. Um, and that was what was eventually approved by the FDA and EMA, and commonly known as Luxturna now. It is the first gene therapy to be approved. And right now, it's still the only gene therapy to be approved in the ocular space. So we're working hard to make sure it's not all by itself. We're trying to get a few more approvals done. So
That's awesome. Yeah, I am familiar with, the name Luxturna, obviously. So wow, I'm actually kind of shocked that my, day off from school went toward furthering Luxturna.
absolutely.
Kind of crazy.
Absolutely. Yeah, so it was
Um,
worth taking a day off of school, huh?
what's up?
So it was worth taking a day off of school.
I mean, I'll take a day off of school to take a nap, so this was even better.
That's good. That's good.
So yeah, it was. I definitely say it was worth it. So how was you know after kind of getting those endpoints that you were looking for and getting the data, what was the next sort of challenges to get to the endpoint of Luxturna that maybe some of these other projects for vision research are going to face as they come down the pike as well?
Yeah, I think one of the things was the idea that because there really wasn't a blueprint of how to run things or do things, we were really devising our own methods, and with the support of, the whole team, and input from the regulatory agencies, we were eventually able to go to the FDA and have an advisory board, and based on the totality of the data, they found that it was a beneficial product that was had a good safety profile as well, and eventually was approved. So that was back in 2017, and unfortunately, we haven't had another approval in the gene therapy space for the eye since then. And some of the issues has been really in endpoints and knowing what the right endpoint is for the right disease, because it's not a one size fits all, unfortunately. And so there's still a lot of work in trying to understand what endpoints our regulatory colleagues would like to see in clinical trials. And the other thing is the design of clinical trials. Things evolve over time. At the time, we were one of the first to develop, but more and more, as we start to learn, as regulators start to learn, and other folks in the community start to learn, things change over time. And so, I think there's still some talk about what is the optimal design, and then the idea of harmonization between the U.S. and maybe the EU and the regulatory aspects. And then, of course, a lot of these are very rare diseases, and so. Unfortunately, we need funding to run clinical trials. Unfortunately, it can get quite resource heavy. And so that's a constant need, is to fundraise and make sure that people are still very interested in rare disease and the potential that gene therapy and other genetic therapies could actually bring to the community. So those are some of the challenges that we are facing at this point, and we're getting through and we're working our way. And there are a lot of groups that are working on a lot of different products, both in the gene therapy space and in small molecule and other things as well. So we're hopeful that will be able to impact the community with some positive therapeutics, hopefully in the near future.
Good, that's awesome. What do you think? I mean, maybe there isn't one, but if you had to guess, or if you maybe know of something, what could be the next big breakthrough? Kind of what's the next Luxturna to happen here in the
Well,
2020s? Do you think?
yeah, that's a very good question. I mean, there are a lot of great companies that are working on different diseases. Right now, I would think X-linked RP, retinitis pigmentosa, has a number of folks going after that target. There are some folks that are working on something called retinoschisis, and just a number of different products. Of course, gene therapy has also branched out into more common diseases such as wet AMD, and people are working on that as well. So there are a lot of things that people are working on, and there's a lot of work going on in the basic sciences as well. Is how can we deliver genes a little better than what we're doing? How can we make them a little safer, and decrease the potential inflammation that may happen? So I would say right now there's work on all different fronts. The work is definitely on the preclinical and getting things where we have evidence to go into human trials, and then obviously the human trial itself and what the endpoints are, the best design, and of course we're always looking at patient safety as the first and foremost thing that we're concerned about.
Sure.
And then, of course, we're working on the regulatory aspects too, as to what regulators want to see, and maybe we need to inform them a little more and let them know some of the things that we see in the ocular space that we feel might be a good endpoint. So I think there's all that work. There's even some work in the commercialization aspects as well. And of course, throughout all this time, there's the work on fundraising and how we can get to different kinds of investors, whether they're private investors or philanthropic or something of that nature, to make sure that we have the resources to continue these programs.
Awesome. How about you? What's kind of been your focus in recent years and going forward here? What have you been working on?
So I left Spark about a little over three years ago, and I joined a small company called Sparing Vision. It deals in what we call gene-independent approaches. So
Hmm.
instead of dealing with, in a lot of genetic medicine, you're dealing with one specific gene, and you're basically trying to replace that gene. Unfortunately, with this approach, a lot of times it deals with a very small number of patients, which is not a bad thing at all. It's just that we don't have the resources to hit all 300 different genes right now that are involved in inherited retinal disease. So this is just another way, as I like to put it, just another shot on goal that we could possibly help folks with retinitis pigmentosa, at least slow down some of the vision loss by going through a common pathway that we see as a neuroprotective pathway and agent
So how
some.
does that work? Is that something that deals with the brain rather than the eye?
Well, it's an interesting thing is that our founders of the company, Sparing Vision, was Professor Jose Sahel and Thierry Livellard, who were at the time at the Vision Institute and at the National Eye Hospital in Paris. And they basically tried to identify a factor that they thought would be in the eye that protects the central vision cells. Because in RP, or retinitis pigmentosa, the outer cells, or the cells in the periphery, tend to degenerate first, and that's usually directly caused by the genetic defect that happens. And but there wasn't a direct link to the central cells, what we call the cone cells, and that's what we use for color vision and central vision, daytime vision. And they found a factor that was actually made by those rod cells. And so that factor helped those central cells stay alive. And this happens in all of us.
Hmm.
And the idea in these RP diseases is that those rod cells, or those peripheral cells, die and they stop making this protective factor. And so the idea that is maybe one of the reasons why these cones die. So if we restore that through gene therapy, maybe we can at least save those central cone cells from dying, or at least slow it down, so that people with retinitis pigmentosa could potentially have a more usable vision, and not go to significant vision loss centrally as quick as they do, and just prolong that. And that is through that common factor of RDCVF working on a different mechanism. So So we're looking at that, and we're in clinical trials now, and we'll see how it goes.
That's fascinating, and specifically on a personal note for me, because I guess I'm weird. The best vision I have is peripheral. All of my
Sure.
central vision's gone. The only stuff I have, and working in the obstacle course, and
Yep.
the only thing I have is the peripheral. So perhaps is there some weird reason that my rods didn't die fast enough or something?
Yeah, because a lot of times the different diseases will target your cones first, and they'll have genetic variants that cause dysfunction in the cones preferentially over the rods. We're also working on another program that is still in the preclinical stage, but it's at least in the preclinical realm, it actually restores what we call dormant cone cells, so cells that aren't functioning, but they're still present.
Hmm.
And we may be able to restore what we call phototransduction, and that's basically where the light coming into the eye is turned into an electrical response, and that's sent to the brain, and the brain tells you that you see. And so maybe we can restore that in folks that have lost their central vision as well. But that's still preclinical, and there's still a lot of work to be done on that. But if everything goes well, maybe we'll be able to start a clinical trial next year sometime.
I mean, all of this is fascinating. Okay, last few questions that I ask every single guest here. Number one, what do you think are the biggest bright spots all across the field of vision research going forward in the next, like 5, 10 years? What do you have your eye on coming down the road?
So I think there's a lot of things going on. Obviously, gene therapy, whether that's single gene augmentation, like we see with Luxturna, or gene independent, like we're doing. There are gene modifiers that are also being looked at. There are something called optogenetics, which is taking cells that aren't normally photosensitive to a high degree and making them photosensitive. So there's a lot of work in that as well. There's other types of genetic modalities coming down the pike. Gene editing, we've all heard of CRISPR. So that's getting some traction as well in the eye. There's also something called antisense oligonucleotides, which actually acts on the RNA level instead of the DNA level. So it's just a different way of using genetics to kind of do things. And then, of course, with gene therapy, there's always the talk of cell therapy as well. And so that is also being looked into right now. There are a couple programs in the clinic that are looking at cell therapies. So, as I like to say, we just want as many shots on goal as possible. And wouldn't it be great that one day that folks with ocular diseases will have multiple options to choose from instead of being at a place right now where we don't have a lot of options. So
Absolutely.
that's what I think. There's a lot of things coming down the pike.
Cool. That's awesome. And then, obviously, this field is not easy. This field is, very chaotic. The biggest hurdles, that you think the field as a whole will have going forward the next few years, what do you think those? Sorry, my
I think
dog
the
is.
challenge.
I
Yeah,
just
no,
jumped out of my
totally.
seat. My dog is not
Oh,
happy. We have a car going
uh-oh.
by. Yeah,
Oh, I see. Um, I do think that some of the challenges that we have right now is the whole idea of making sure that we have the right endpoint for the right clinical trials and the design of the clinical trials, that we have more collaboration with the regulatory agencies. And above all, it's resources, fundraising, making sure that even big pharma and biotech stay interested in the rare disease space and investors and things of that nature. So those are some of the things that I see are some of the challenges ahead. But it's an area that has high unmet need. We really need to get some therapies in here to help folks. And so hopefully we will have more approvals in the near future.
awesome. And then final question. Also, I apologize to anyone who, if that if sassy hurts your eardrums a little bit. That was
No worries,
I
no
jumped
worries.
out of my seat, so I know it was quite uncalled for. But she, a blade of grass twitches in the yard and she loses her mind. So I think
That's great
everyone with a
hearing.
dog knows how that goes. Final question though, for our folks watching, what can we be doing to help this along to kind of clear some of these hurdles? What can people do each and every day to help this along?
Well, I think organizations like the CRB1 Foundation are integral. The patient voice is really just a key part of the whole drug development pathway, and that really helps us to understand what the right endpoints are. It helps us to understand what the patient journey is like, what the issues are in their vision and and how we can actually help that. And so I do think that patient organizations are vital in the whole process. I think the best thing for folks is to, if there are natural history studies, they can be a part of that, because those are really vital for us to understand the way the disease goes over time. If there are registries, that if there is a gene therapy or if there is a product that's out there and they're looking for clinical trial participants, that there is a way that we could not necessarily us, but hospitals and our sites contact them to let them know that these trials are ongoing and things of that nature. So networking is great, but also it's just to stay really involved. We at Aspiring Vision and when I was at Spark, we always tried to incorporate the patient voice in pretty much everything we do when it comes to the clinical end of things. So, so you know, key key to the whole process. And obviously, I know your mom pretty well, and she's a mover and a shaker, so we always appreciate her participation. So
That's one thing. She's also a, also a shepherd of dogs that I
there
guess,
you go.
getting them out of the room and everything. Also a producer of this great show. So want to thank you, Kristen Smedley, once again for helping out today. And a huge thank you to Dr. Chung. We really appreciate your time today. And thank you to everyone else at home, wherever you may be, however you may be watching. Thank you so much. We really appreciate it. This has been yet another episode of Eyeing Progress with Dr. Dan Chung, and we could not thank him enough. And we hope to see you here next time on the Curing Retinal Blindness Foundation YouTube page for another episode of Eyeing Progress. I'm going to get through this outro, I swear. I'm Mitchell
No,
Smedley. See you next time.
thanks again.